Okay, so welcome back to this next video in which we are discussing rheumatoid arthritis and anti-rheumatoid drugs. Okay, so in the previous video what we discussed is the leading theory for what causes the autoimmune reaction which underlies rheumatoid arthritis. Okay, and we saw that the leading theory is that you initially get infected with some pathogen, okay, and you then launch adaptive immune responses against the antigens of this pathogen, and a certain epitope of one of those pathogens, which has an adaptive immune response initiated against it, turns out to be too similar to an epitope of a self-protein. Okay, and this means that the adaptive immune response that you launch against that epitope of this antigen, which we'll call an arthritogen, is then going to be able to attack that self-protein, that autoantigen. Okay, and it seems that that autoantigen then is present within the synovial joints, and that's why the adaptive immune cells infiltrate into the subintima of the synovial membrane of the synovial joints. Okay, and we seem to know what property the arthritogen and the autoantigen share. It seems to be that the arthritogen has a citrulline residue in it. Okay, so it's a citrulinated protein, and the autoantigen, whatever that is in the synovial joints, that also seems to be a citrulinated protein. Okay, so you seem to be launching an adaptive immune response against an epitope that contains a citrulinated, a citrulline residue. Okay, and then that causes you to break the immune tolerance for these citrulinated proteins that seem to be present in the synovial joints. Okay, right. So what we now want to turn our attention onto is a discussion of the anti-rheumatoid drugs. Okay, and the way we're going to structure this is we're going to start off by discussing the drugs which are the first-line treatments for rheumatoid arthritis. Okay, so we're going to discuss the NSAIDs and the DMARDs first, okay, because usually they are used in combination together as the first-line treatment for rheumatoid arthritis. Okay, then we're going to talk about the glucocorticoids, which can also be used to treat rheumatoid arthritis. And then finally, what we'll talk about is the biopharmaceuticals, which are more effective than the DMARDs in preventing the progression of the disease. However, they are extremely expensive, and therefore they are a second-line therapy for people in whom DMARDs are ineffective, basically. Okay, right, so anti-rheumatoid drugs used to treat rheumatoid arthritis. Okay, right, so first of all then, we are going to discuss the NSAIDs. Now these are not used on their own to treat rheumatoid arthritis, because all they treat is a little bit of the inflammation. They'll make the swelling go down a little bit in the rheumatoid arthritis joints, okay, and they also treat the pain. However, they do nothing really to stop the autoimmune response, and they don't stop the joint destruction, okay, which is why they need to be given in combination with drugs that better attack those things, okay, such as the DMARDs. But we'll start by discussing the NSAIDs and what they're going to do. Okay, right, so firstly, what does it stand for? Well, it stands for non-steroidal anti-inflammatory drug. So the N is for non, the S is then for steroidal, okay, the AI is for anti-inflammatory, okay, and the D is then for drugs, so non-steroidal anti-inflammatory drugs. And I will give you a few examples of non-steroidal anti-inflammatory drugs before we talk about how they actually work to reduce the inflammation in rheumatoid arthritis and also the pain. Okay, so examples of non-steroidal anti-inflammatory drugs, and these are all quite famous drugs. So ibuprofen is an example, aspirin is another example, okay, diclofenac and indomethacin are also two other examples. So diclofenac, that should be a C there. Okay, I'll try and turn it, now I don't know what it looks like, but that should be a C. Okay, diclofenac, and then also indomethacin. Okay, right. So, how do these drugs work? Well, basically, they work by stopping the production of prostaglandins. Okay, so if I just get up my sheet back from the pathogenesis of rheumatoid arthritis, okay, remember that once you've got this infiltrate of adaptive immune system cells that results in the production of interferon gamma and immune complexes in the extracellular fluid of the synovial membrane, that triggers the type A synoviocytes, the synovial macrophages, to start releasing a bunch of pro-inflammatory molecules. Okay? And two of these that we looked at were prostaglandins. Prostaglandin I2, also called prostacyclin, which is pro-inflammatory. Okay, that causes vascular smooth muscle cells surrounding the arterioles to relax and therefore causes vasodilatation of the arterioles, which increases blood flow to the affected area basically. So that's pro-inflammatory, that's causing in part the swelling in the joints. Okay, prostaglandin E2 then, the second of the prostaglandins here, this was very important in triggering pain. This acts on pain neurons in the synovial joint. Okay, and there is good evidence now that prostaglandin E2 effectively sets up rhythmic firing of those pain neurons, similar to what you have in the heart basically, where they just fire continuously at a certain rhythm. Okay, right. So basically the non-steroidal anti-inflammatory drugs are going to work by stopping the type A synoviocytes, these synovial macrophages, from producing these two prostaglandins, and therefore are going to help with the swelling associated with rheumatoid arthritis, and they're also going to help reduce the pain associated with rheumatoid arthritis. Okay, so how do they stop the production of the prostaglandins? Well, to understand that, we need to know something about the production of the prostaglandins. Okay, so prostaglandins, both prostacyclin and prostaglandin E2, are made from the starting molecule arachidonic acid, okay, which is often just abbreviated down to AA. Okay, now in order to make final prostaglandins like prostaglandin I2 and prostaglandin E2, what you firstly have to do is convert arachidonic acid into something called prostaglandin H2. Okay, and then you can convert prostaglandin H2 into the final prostaglandins such as prostaglandin I2 and prostaglandin E2. Okay, now, how do you convert arachidonic acid into prostaglandin H2? And it's this conversion that the non-steroidal anti-inflammatory drugs are going to stop. Okay, well basically arachidonic acid is firstly converted into an intermediate called prostaglandin G2, PGG2, and then it's converted on to prostaglandin H2. Okay, now these reactions are so important that they actually have their own names. Okay, so this first reaction, which is the conversion of arachidonic acid into prostaglandin G2, this is called the cyclooxygenase reaction. Okay, then the second reaction, which is the conversion of prostaglandin G2 into prostaglandin H2, this is called the peroxidase reaction. Okay, and both the cyclooxygenase reaction and the peroxidase reaction, which overall achieve the conversion of arachidonic acid into prostaglandin H2, these are both catalyzed by the same enzymes. Okay, so both these reactions here and here are catalyzed by enzymes called cyclooxygenase enzymes. Okay, and for short, cyclooxygenase enzymes are called COX enzymes. Okay, C for cyclo, ox for oxygenase. Okay, and there are two main forms of cyclooxygenase enzymes. There's cyclooxygenase 1 and cyclooxygenase 2, and they're both capable of catalyzing both of these reactions basically, and therefore they're both capable of converting arachidonic acid overall into prostaglandin H2. Okay, right. Now, the non-steroidal anti-inflammatory drugs, ibuprofen, aspirin, diclofenac and indomethacin, they all work by inhibiting the cyclooxygenase enzymes. Okay, so the NSAIDs they work by inhibiting the cyclooxygenase enzymes, and therefore you can no longer convert the arachidonic acid into the prostaglandin H2, and if you can't do that conversion, then you can't make prostaglandin I2 and prostaglandin E2 anymore, because they need the prostaglandin H2 in order to be converted into them. Okay, so you can't just go from arachidonic acid to these without first going through this conversion. So that's how the non-steroidal anti-inflammatory drugs prevent the synthesis of prostaglandins by those synovial macrophages. Okay, and the stopping of the production of prostacyclin is what produces the reduction in the swelling of the joints. Okay, and the stopping of the production of prostaglandin E2 is what causes the pain relief associated with these drugs. Okay, right, so those are the non-steroidal anti-inflammatory drugs. Now, as I say, they do nothing to stop the destruction of the joints, okay? They do nothing to stop the underlying autoimmune reaction, okay? So all they do is reduce the inflammation slightly and reduce the pain, okay? So they are not given on their own, basically. The first line treatment for rheumatoid arthritis is to give a non-steroidal anti-inflammatory drug with what's known as a disease-modifying anti-rheumatic drug. Okay, and for short, these disease-modifying anti-rheumatic drugs are abbreviated to DMARDs. Okay, and it's these DMARDs that we now want to study. Okay, so firstly I'll write out their full name in full. Okay, so DMARD stands for disease, that's the first D, and then it's disease modifying, that's the M, and then AR is for anti-rheumatic, okay, so anti-rheumatic or anti-rheumatoid, whatever, and then drugs. Okay, so disease modifying anti-rheumatic drugs, that's what DMARDs stand for, and there are quite a few of these. Okay, we're going to start by discussing the main one, and then we're going to work our way through the others, and a lot of them have quite similar mechanisms of action. Okay, right, so the first one that we are going to discuss is going to be methotrexate, okay, which is the most common one that is used. Okay, methotrexate is really the first line treatment for rheumatoid arthritis. Okay, and the way that it works is by producing immunosuppression. Okay, by stopping that autoimmune response that is actually driving the disease. So methotrexate for short is abbreviated to MTX. Okay, right, so how does methotrexate then produce immunosuppression? Well, basically the way that it produces immunosuppression is by stopping cell proliferation. Okay, so methotrexate stops cell division. Okay, and for this reason methotrexate is also used as an anti-cancer drug, where clearly stopping cell division is very useful. Okay, now, why does stopping cell division then produce immunosuppression? Well, you'll remember, if you remember back to our discussion of the adaptive immune response, and I'll just get my page for the adaptive immune response back up. Remember, as part of the adaptive immune response, it was very important that the T cells could expand, basically, and also that the B cells could expand. Okay, when you activated the CD4 positive T cell here, and the first thing it did was it divided and divided and divided to produce a huge, great population of identical T cells, and then these can all go on to differentiate into T helper cells. Okay. Similarly, when we activated B cells, what did they do? They clonally expanded. They produced loads of identical B cells, which could then differentiate into plasma cells. Basically, methotrexate is going to block these cells being able to do this, okay? And that's going to completely shut down the immune response, okay? Because one B cell or one T cell isn't going to be able to do anything. It has to be able to clonally expand like this to have a non-negligible effect, basically. Okay, so by stopping cell division systemically in the body, you can completely stop the immune system from functioning, basically. Okay, and that's how methotrexate is an immunosuppressant. Okay, right. Now, why does that help in rheumatoid arthritis? Well, fundamentally, what is driving the rheumatoid arthritis? It's this continued autoimmune process. If you got rid of those CD4 positive T cells, those B cells and those plasma cells, which were infiltrating into the subintima of the synovial membrane, then the whole thing stops, basically. They are what are driving the entire process. Okay, so that's why shutting off the adaptive immune system is going to help there. If we stop any further division of those adaptive immune system cells, then eventually the immune system cells will die. They don't live forever. Okay, white blood cells are being continually turned over in the bloodstream. Okay, so all you then have to wait for is them to be gradually cleared, and then you're not going to be making any more now because we've stopped cell division, and therefore you will get an end to this autoimmune process is the idea here. Okay, right. So what now remains to be seen then is how does methotrexate actually stop cell division? How does it stop cells dividing? Okay, well, basically, it works by blocking the synthesis of a whole bunch of nucleotides that are needed for the synthesis of DNA and the synthesis of RNA. Okay, now, the first thing I want to discuss is why blocking the synthesis of DNA and blocking the synthesis of RNA is actually going to stop a cell dividing, and then we'll look at how methotrexate actually stops the production of certain nucleotides that are needed to produce DNA and RNA. Okay, so if we think about cell division, if we think about one cell splitting into two cells, what are the essential things that have to happen when one cell divides into two cells? Well, firstly, you have to copy the entire genome. That's one of the obvious things that you have to do. Okay, here this cell has one copy of the genome, one copy of all 46 chromosomes. Okay, now, if we've got two cells, each of them has one copy of all 46 chromosomes, and therefore you need to copy the entire genome. That's going to require a huge amount of DNA synthesis, basically. If we've blocked the production of nucleotides that are needed for DNA synthesis, then we're not going to be able to copy the genome, and therefore we're not going to be able to divide. Okay, but there's more to it than that. Okay, also, if a cell wants to divide into two, it has to duplicate all of its essential proteins. All of the proteins of the cytoskeleton are going to have to be duplicated because both cells are now going to need a cytoskeleton. All of the essential enzymes of respiration, all of those are going to have to be duplicated because you're going to need enough for two cells rather than one. Okay, all of the proteins of signal transduction and all of that, they're going to have to be duplicated as well. So you're going to have to make a huge amount of protein, basically, when you want to divide into two. Okay, to synthesize protein, the central dogma of biology says that you have to firstly make mRNA. Okay, if you haven't got the appropriate nucleotides that you need to build mRNA, then you're not going to be able to make more protein. Okay, so you also block protein synthesis by stopping RNA synthesis. Okay, so the two things that are going to be blocked basically by this drug are DNA synthesis, okay, which is going to stop the copying of all of the chromosomes, okay, so it's going to block DNA replication, and you're also going to block protein synthesis by blocking mRNA synthesis, okay, and if you haven't got those two processes, then the cell just simply cannot divide, basically. Okay, right, and you can't make DNA and mRNA because you can't make some of the nucleotides that you're going to need in order to make the DNA and the mRNA. Okay, so now let's look at the details of why methotrexate actually achieves this blockade of production of certain nucleotides. Okay, so it works by blocking an enzyme called dihydrofolate reductase. Now let me tell you what dihydrofolate reductase is involved in doing. Okay, so basically dihydrofolate reductase is involved in turning folate or folic acid into firstly dihydrofolate, which I'll abbreviate down to DHF, and then into tetrahydrofolate, which I'll abbreviate down into THF. Okay, so I'll just now put these names in full. So DHF stands for dihydrofolate. Okay, and then you can imagine that THF stands for tetrahydrofolate, so replace the di with tetra. Okay, and actually dihydrofolate reductase doesn't just catalyze the conversion of dihydrofolate into tetrahydrofolate, it actually catalyzes both of these. Okay, so this enzyme, which is usually abbreviated down to DHFR, okay, which is dihydrofolate DHF reductase, which is the R. Okay, this enzyme catalyzes the conversion of folate into dihydrofolate and then dihydrofolate into tetrahydrofolate. Clearly it is named because of catalyzing dihydrofolate to tetrahydrofolate. Okay, so the name dihydrofolate reductase describes what it's doing in this conversion, but remember it also catalyzes this conversion that occurs before that. Okay, right. Now, tetrahydrofolate is needed to catalyze a certain reaction that is needed to synthesize certain nucleotides, basically. Okay, and let me now describe the reaction that requires tetrahydrofolate to work. Okay, so the reaction that requires tetrahydrofolate to work is the conversion of deoxyuridine monophosphate into deoxythymidine monophosphate. So let me draw some pictures for this. Okay, so deoxyuridine monophosphate basically just means deoxyribose with a uracil organic base attached to it and then a single phosphate group attached onto that. Okay, so here is the uracil organic base, here is the deoxyribose with its fifth carbon coming up here, and here is its phosphate group. Okay, so this is representing deoxyuridine monophosphate, okay, which for short is abbreviated down to DUMP. Okay, so deoxyuridine monophosphate is usually abbreviated down to DUMP, like so, dump. Okay, so uridine, let's talk through what this name actually means. Uridine means a ribose sugar with uracil bound to it. Okay, so the organic base uracil, which is used in mRNA. Okay, so here is uracil. Okay, then if you attach uracil to a ribose sugar, that will create you uridine, but we don't want uridine, we want deoxyuridine, and therefore you need to attach the uracil organic base to a deoxyribose sugar rather than a ribose sugar. So you need to attach it to the type of ribose sugar that's used in DNA rather than RNA. And the type of sugar used in DNA is deoxyribose. Specifically, it's 2' deoxyribose, which means that there's no alcohol group coming off the second carbon of the ribose ring here. Okay, so that's 2' deoxyribose represented by that green pentagon there. Okay, and then it's going to have a single phosphate group coming off this, okay, which is that purple blob up there. So this now represents deoxyuridine monophosphate, DUMP. Okay, uracil bound to 2' deoxyribose gives you the deoxyuridine, adding on a single phosphate gives you the monophosphate. Okay, now deoxyuridine monophosphate can be converted into deoxythymidine monophosphate. Okay, which is just now the same molecule, but you've converted that organic base from uracil to thymine, basically. Okay, so thymidine means thymine bound to the ribose sugar, basically. Okay, deoxythymidine then means thymine bound to the deoxyribose sugar. Okay, so let me put this in here. So here is our deoxyribose sugar again, which we haven't changed. Here is the phosphate group, and now we've got the organic base thymine attached to that deoxyribose sugar there. Okay, so in green here, this is our deoxyribose sugar once again. Okay, here is our phosphate group coming off the alcohol group that's attached onto the fifth carbon of the deoxyribose. And now, instead of uracil, we've got thymine there. Okay. So we have converted deoxyuridine monophosphate into deoxythymidine monophosphate, or DTMP. Okay, and for this conversion you require tetrahydrofolate. Okay, so what catalyzes this reaction? Well, this reaction is catalyzed by thymidylate synthase. Okay, so thymidylate synthase, and that name refers to the fact that deoxythymidine monophosphate can also be called thymidylate. Okay, now when thymidylate synthase turns deoxyuridine monophosphate into deoxythymidine monophosphate, it uses a molecule of tetrahydrofolate up, and the molecule of tetrahydrofolate is converted back to dihydrofolate. Okay, so what I'm saying here is every time you convert a deoxyuridine monophosphate molecule into a deoxythymidine monophosphate molecule, you also use up a molecule of tetrahydrofolate, which is converted back to dihydrofolate here. Okay, so to synthesize deoxythymidine monophosphate, you have to have tetrahydrofolate. Okay, and then to convert the dihydrofolate back to tetrahydrofolate to replenish our stores, you require this enzyme dihydrofolate reductase. Now, deoxythymidine monophosphate is then needed to produce all sorts of nucleotides, basically, okay, including thymine nucleotides. Well, that one's obvious, okay, but also less obvious, it's also needed to produce purine nucleotides, okay, so adenine and guanine nucleotides. You can convert this molecule into adenine and guanine nucleotides. Okay, so basically, if you block this reaction, okay, if you block the conversion of deoxyuridine monophosphate into deoxythymidine monophosphate, you block the synthesis of thymine nucleotides for both DNA and RNA. Again, it's not instantly obvious that you're doing it for RNA as well, since we're working with deoxyribose here, but actually you need this reaction to make the RNA nucleotides as well. So you block the synthesis of DNA nucleotides for RNA and DNA of thymine, adenine, and guanine, okay? And therefore you really cannot make DNA anymore or RNA without those nucleotides, okay? So how are we going to block this reaction? Well, we are going to block it by inhibiting the dihydrofolate reductase. This is the target for methotrexate. Methotrexate inhibits dihydrofolate reductase and therefore stops the conversion of dihydrofolate into tetrahydrofolate, and also stops the conversion of folate into dihydrofolate. But it's this conversion that's really important here, because remember, every time this reaction is catalyzed, you convert tetrahydrofolate back into dihydrofolate, okay, and therefore really it's this replenishing reaction here that you need to be operational, otherwise you're going to run out of tetrahydrofolate, okay, and if you inhibit the dihydrofolate reductase enzyme with methotrexate, then you stop it, okay, you do not produce any more tetrahydrofolate, you run out of tetrahydrofolate, that blocks this reaction from proceeding, you block the synthesis of adenine, guanine, thymine nucleotides, you block the synthesis of DNA and RNA, and therefore you completely block cell division. Okay, so that's the basis of how methotrexate is functional against rheumatoid arthritis, by blocking cell division, thereby producing a very powerful immunosuppressive action, okay, and hence stopping the autoimmune response which drives rheumatoid arthritis, okay, and therefore leading to remission from the disease. Now, methotrexate therapy usually takes 1 to 2 months to be effective, okay, so I'll put that up here. So this takes usually 1 to 2 months, and that's because, after all, you have already got loads of T helper cells, you've already got loads of B cells, loads of plasma cells, okay? You take this drug, it stops cells dividing, okay, but it doesn't get rid of the cells you've already got. You have to wait for those cells to gradually be removed from the blood, okay? They don't live forever, they will turn over, so you will gradually lose them, okay? You won't make any more if you're taking this drug, and therefore you have to wait for the ones that you've already got to go away, and before you actually see the benefits of this drug. Okay, so that's why it takes quite a long time to work, but it is incredibly effective at producing remission from rheumatoid arthritis. Okay, right, so we'll call it there for this video. In the next video, we'll discuss more immunosuppressants that can be used to treat rheumatoid arthritis.